So today we are changing a little bit the subject, um, and we were talking about networks and the analysis of the network structure and quantifying things on biological networks. But there are some questions that we cannot address only with the static pattern of interactions in networks. So we need to have a more complete uh, description of the system that we want to study in order to make some inferences about how it works or why it is designed in such a way. So we are going to start by introducing this part of it's written modeling in biological systems, but it should be dynamic modeling in biological systems. There are many different types of models, and exactly we are starting by that. So everything that you do in science somehow implies modeling. Because you cannot do experiments if you don't have a previous model, a working hypothesis of how the system works. Then you make predictions and then test those predictions. So in order to design experiments, to validate or to test some hypotheses you have about the system, you have to have a model, and that model is based on those hypotheses that you have about the system. So a model, there are many different types of models, but there are even, for example, people in the lab use experimental models like model organisms and stuff like that. But the model is always a simplified representation of a real system. And why do we like models? Because they are more easy to play with, and we can change stuff in the model more easily and see what happens. So basically, we can make virtual experiments with models. And that's from that virtual experiment cycle. So we make, if we assume that the model works like that, then if I do this would happen, and I can try it in this simplified model, and then make experiments in a system that is closer to the real system that we want to study. So these play around with the model will facilitate our understanding of how the real system works. Here is an example that I find it's a very nice example. That is, this is a physical model of construction of houses in Lisbon after the earthquake in 1755. Actually, from what I read somewhere, after the earthquake, it was a big earthquake, and we got help from people from abroad. So the engineers from different countries, and actually, I think some Dutch engineers came, and they tried to develop a system to construct buildings that would be more resistant to the earthquakes. So that was a technological advancement that occurred after that earthquake. And they built physical models like this one. So this is a small scale model of the building, and they simulated earthquakes. So they constructed these models on top of plaques, and then they shaped those plaques to simulate the earthquake and evaluate if the different types of building systems were more or less stable and robust to the perturbation of the earthquake. So this is also a type of model, it's a physical model. In biology, particularly in molecular and cellular biology, people work mainly with conceptual models. So normally people studying a pathway, a cellular process, or an interaction between cell types or whatever, they have in their heads, and when they design the experiments, they have some kinds of a cartoon like this in their heads that allows them to think about the system. So these conceptual models identify what are the components of the system, what are the interactions between those components, what are the external variables that may affect the system behavior, and eventually also what are the outputs of the system behavior. And then you can do, even without any mathematics, you can do virtual experiments. So you can ask, if I take this element out, so this pi3k, if I take him out, what will happen? In this case, maybe it doesn't happen that you don't see that that much, because you have a parallel pathway that seems to do exactly the same, for example. So you can make virtual experiments with these conceptual models, and if they are simple enough, you can do that even without a mathematical model. But these models are always simplifications of the real system, and you can have different levels of simplification. And sometimes to address some questions or some to try to solve some scientific problems, you have to address, you have to include more components in your system, or you have to assume that there is a more complex environment affecting your system behavior. So in models that are more complex, and by more complex, it means that it has either it has more components, but having more components is not necessarily implies that the model is complex, because if you have more components, but all components behave similarly, have a very similar pattern of behavior, so you can more easily extrapolate from the behavior of one component what the others will do. So it's not that complex. But if you have more complex and more complex patterns of interaction, so more interactions, and you can have, for example, one thing that makes it really difficult to make predictions just in your head about the system is when you have feedback loops. So when you have there is a signal, external signal that affects some components, but at some point there are some of these components that signal back to other components that influence the origin of the signal. And you can have feedback cycles, feed-forward cycles. Those feedback cycles or feed-forward cycles can be positive or negative. And it makes it difficult to make predictions just by imagining in your head what would happen if I increase this activity or decrease that activity. So you get to a point where if you want to work with larger, more complex systems, you need to have some kind of computational or mathematical model to allow you to make predictions of how the system behaves after some kind of perturbation. So that's really a must when you study complex systems. Traditionally, what people have done is they study the system by parts. So they focus they want to understand this complex system just by studying one bit at a time, and that is the reductionist approach. Experimentally, it makes sense because you can make more controlled experiments, but as we spoke before in the first lesson, I think you cannot, when you study just the parts individually, it's difficult to predict the behavior of the whole system just by assembling the parts, because there are some behaviors that emerge from the patterns of interactions. So we need to build and analyze mathematical models, and there are different types of mathematical models. And in this lecture, and probably in the next one, we are going to discuss the most common type of mathematical model to describe the dynamics of biological systems, that are mathematical models based on systems of differential equations. But these are not the only approach, and in the In this course, we will also talk about some alternative approaches. So, but basically these mathematical models, what they do is that they integrate available knowledge and hypotheses about the system. So when you build a model, you are encoding in mathematical language what is known about the system presently, and if what you know about the system is correct, then the predictions of the model should agree with experimental observations, and that would allow you to make even further predictions in different conditions that will allow you to test and validate the model and check. The idea is that you keep making new predictions until the predictions fail, because it is when the predictions fail, so they don't, the predictions are not able to reproduce experimental observations, that you know that you need to change something in the model. Okay, so normally we have to start from the conceptual model. We have to identify in that conceptual model what are the components that we want to include in our model. Some of those components are going to be dependent variables. And these dependent variables, we call it dependent because their behavior is going to be defined by the model equations. So actually what we can predict is the behavior of those dependent variables. But normally we have to include also some independent variables, that is variables that are not, whose behavior is not going to be predicted by the system, by the model, but their value is important to define the behavior of the dependent variables. And then we want to write some kind of mathematical expressions, the equations. Normally, if you want to be able to predict the evolution in time of the dependent variables values, you write differential equations. If those values, there are other options. For example, there are difference equations, but it is more common to see models with the differential equations, mainly because we are working with the variables that have continuous values. So normally the variables that we want to study as dependent variables are the concentration of some molecules in our system, and that is a continuous variable. And as we have previous knowledge of chemical and biochemical kinetics, we have some expressions that we are going to talk later that allow us to easily write those systems of differential equations. So and those chemical and biochemical kinetics were developed using mainly differential equations. And also the time is also continuous. Some question?
Yes, please. Could you explain again what makes a variable dependent?
So this is just the dependent variables are the ones that their value through time is going to be predicted by the model. So each dependent in a model of differential equations, each dependent variable is going to have its own differential equation. And the independent variables have values that are set a priori of building the model.
And they will not be updated or...
Yes. Yeah. For example, we can define that there is an independent, for example, if we are modeling the evolution of plant metabolism during the day, and that metabolism is affected by light intensity, for example. So the light intensity is an independent variable. It's not, it doesn't need to be a constant value, but the value of the light intensity is not going to be defined by the model. We are going to input that variation through time. To be able to... Is that clear?
Yes, thank you.
Okay. So I was talking that we normally use differential equations because most of these biological models of biological systems use rate expressions from chemical kinetics or biochemical kinetics, and those were originally developed in using differential equations, assuming that all the molecules that are interacting and having reactions that transform one into another occur in solutions that are homogeneous in space. So all the molecules flow around freely. And in those conditions, when we have for each species, for each components of the model, we have a large number of molecules. And if we assume that we have an homogeneous space, then the predictions made with the differential equations can be quite accurate. Of course, biological systems are not that so homogeneous in space, and sometimes some of the components don't have a very large number of molecules in the system that we are modeling. So sometimes those differential equations are not a good approximation, and we, for example, may have to use stochastic models. But even for the stochastic models, we can use similar equations. You can translate a system of differential equations into a stochastic one. So these differential equations, there are some classes of differential equations that have known solutions, so that you can, they have analytical solutions. So you don't have to run a computer simulation to predict the evolution of the dependent variables through time. But normally, most of the systems of differential equations that we write to describe the behavior of biological systems are non-linear differential equations. By non-linear, it means that there are some terms of these equations where we have products, for example, that is the most common situation, of different dependent variables in the same term of the equation. For most of those systems, nonlinear systems of differential equations, or systems of nonlinear differential equations, you don't have an easy analytical solution, or it can be even impossible. You may even know that it's impossible to get an analytical solution in some cases. So normally, with what we most commonly do is perform computer simulations. Basically, we use algorithms that integrate systems of differential equations numerically, and those would transform the differential equations into the time courses of all the dependent variables in the system. To build these systems, we have to be able to perform those simulations. We have to have parameter values for the equations, and that is going to be a limiting step in the building of these models. And once you have the model with the most adequate parameters that you can find, then you should make predictions, and predictions that are not normally to define the parameters. You use some data sets that are available that allow you to fit the values of the parameters. But to validate the model, and that is a necessary step for you to trust the predictions that your model produces. You have to validate model predictions, and for that you have to simulate the model in conditions that are not the same as the ones that generated the data you use to fit the parameters. Make predictions in new conditions and compare with new data that was not used to adjust the parameter values. And if you are successful, if you have good enough predictions that are close in value with the experimental observations, then you can trust that your model is well developed, and then make more in silico experiments to generate new knowledge or to test new hypotheses about how the system works. So to give you an example, this is a paper that I find quite interesting. So it studies a very well-known metabolic pathway that is glycolysis, but in this case in a bacteria that is Lactococcus lactis. This is a bacteria with a biotechnological importance in production of food products, for example. And in this case, the group that was studying this bacteria, they were so in this particular case, in this species, there is a regulatory interaction that are in gray in this figure that are not so common across different species. And they wanted to understand the impact of these regulation. Basically, is that most biological biochemical pathways or metabolic pathways have a way to regulate themselves that is some end product of the pathway inhibits some reaction in the beginning of the pathway. That is a classical way for biochemical pathways to be regulated. But in this case, they have this system where a component more or less in the middle of the pathway up regulates or activates reactions further down in the pathway. So they wanted to study what was the relevance, why it was important for this bacteria to have this regulatory interaction. So this is an example of a mathematical model. So we have some independent variables. For example, the glucose availability in the extracellular media is an independent variable. So we are going that variable is not going to be defined by the model. So we have to input the values of glucose on every time step available in the media. It's normally in metabolic models, some of the metabolites are for example, ATP, ADP, NADH. So these are cofactors that participate in hundreds of reactions in a cell. So their behavior is their availability in the cell is not going to be significantly affected only by this pathway. That's a judgment that you have to do, because you could imagine that this was the most active pathway in the cell, and then the values of these cofactors would be mainly determined by the dynamics of this pathway. But in this case, as these metabolites are used in hundreds of processes, they assume that those ATP values would be an independent variable, because its value would not be defined by the flux going through this pathway. And the same for the phosphate groups. And then you have the dependent variables, and here they are numbered. So this X2 would be the glucose 6-phosphate, X3 would be fructose bisphosphate, and so on, okay, until you get lactate in the end. And these equations are written in a way that we are going to talk about later, that is generalized mass action. So mass action is the most common way to write rates of biological processes and chemical processes. It actually comes from chemical kinetics, these laws of mass action. In this case, they are generalized because in classical mass action models, the exponents of each, so in this case, X2 is formed, so glucose 6-phosphate is formed by the conversion of extracellular glucose into glucose 6-phosphate. So the rate at which this process occurs depends on a kinetic constant, a rate constant. And then you have all the other components of the model that influence this rate. And this would be X1, that is the offline glucose input. So the rate, the more glucose you have, the greater will be the flux through this reaction. It will also be affected by its own value if we consider that there is some reversibility that is common, and it will be affected by X5, that is this phosphoenol pyruvate that participates in this reaction. So normally the chemical reactions, their rate is affected by the availability of the reactants for that reaction. And in classical mass action kinetics, these exponents are called the kinetic orders of the reaction relative to each of the reactants. And normally they are integer values, and they are 1 if, for example, for glucose, only one glucose gives origin to 1 molecule of glucose 6-phosphate. So in classical mass action, this G to 1 would be 1. If 2 molecules of glucose would give origin to 1 molecule of glucose 6-phosphate, then it would be 2. That's the stoichiometric coefficient with which the reactant participates in the reaction. That would be the exponent. In this case, in the generalized mass action, there is freedom to the value of the exponent. So it's another parameter that you have to fit, and that will give you more flexibility to the kinetics of the whole system. And each differential equation is composed of a sum of the rates of the processes that make the component minus the rates of that consume or degrade the component. So that's it. In some cases, so this X2 has only one process forming glucose 6-phosphate and only one process degrading it. So you have only 2 terms in the differential equation. But for example, here X4, 2, 3, 4, you may have more processes because and 5, I guess. So 5, for example, 5 has one way out here, the conversion that goes to the first reaction. It can be go back to the previous component, go forward. So you have a lot of processes, and then each of those processes is a term in the differential equation. So they built the model, and they had to compare the model with experimental data. First, to get parameters that would fit the experimental data, and this is getting the parameters is the more difficult part of these modeling efforts, because if you want to have a model that makes accurate predictions, you have to get good parameter values, and they are not so easy to find. You can use optimization strategies to fit the values of the parameters, but for most cases, you are able to fit well some of the variables, but others, eventually, because there is much experimental noise in the determination of the value of some variables, but some variables are more difficult to achieve a nice fit. But so they were happy with the model that made good predictions for several of the variables, not so much for other variables. But they were not still answering the question, why was these regulatory interactions important? So they built a simplified version of the model. So they built this is a different conceptual scheme that is a simplification of this other one. and as a simpler set of differential equations. And in this case, in this simpler model, they did a comparison of a model where this interaction is present with a model where this interaction is not present. So this interaction is present where? In the equation for x4, right? X4 is consumed, so there is a negative, one of the negative inputs is here. So X4 is formed by a reaction that comes from X3. It's consumed by its participation in the first reaction, so it input times X4, and it is consumed in this last reaction that converts X4 to X5. So you have X2 raised to a kinetic order times X4 raised to 0.5. So this X2 represents this interaction. Okay, so as it is an activation, if this kinetic order H42 is positive, that means that if you have a greater value of X2, you will have a greater value of this rate. Okay, so X2 is activating this reaction. So you can model the presence or absence of this interaction by changing the value of this kinetic order. If this kinetic order is 0, that means that x2 raised to 0 will be 1. And so the rate of this reaction is only determined by the availability of x4, the reactant. But if H42 is greater than 0, then this reaction will be activated by the value of x2. So with this, setting up this more easy to play with model. So these are two models, but this one is, as it has less components, the reactions are more simple, and you have a smaller number of parameters. You can do a more extensive simulation studies. And in this case, they found a condition where they could argue why that regulatory interaction was important. So in these two plots, A and B, the two simulations are shown. And in this first case is the simulation of the system with the interaction present. So where that parameter H42 was greater than 0. And in this simulation, the interaction was not present. So that parameter was H42 was equal to 0. And what they saw is they simulated a situation when there was glucose available. So the input was available in the media at a steady value, a constant availability of the input. And at some point here at t equals 60, no, at t equals 10, glucose is vanishes from the media. So there is no glucose anymore, and this simulates a kind of fasting experience. So you take the nutrients out of the media, and you simulate how the pathway behaves during that fasting period. And if you have fasting forever, so the bacteria will die, of course. But if at some point in the future, so 50 units of time later, glucose appears again in the media, then the system recovers, but it recovers with different kinetics. So in the situation where you have the regulatory interaction, what happens is that once glucose disappears from the media, so this is not present anymore, so you stop producing X1, X2, and so on. And the problem is that if you diminish X2, what happens is that you don't, and this regulatory interaction is present. When X2 goes down, the rate of this reaction is inhibited because it's no longer activated by X2, or the activation is smaller because X2 is getting smaller. So you have a slower rate of the conversion of X4 to X5, and that maintains X4 with a higher level. Okay, and all the other components go to very small values, but X4 is still there. It's going down slowly, so there is some X4 being converted to X5 slowly because the reaction is inhibited because X2 is not present. X4 is also not used with the input because the input is not available. So it's only going to be converted to X5 slowly. If the fasting would last forever, this would go eventually to 0, and that would be equivalent to the death of the system. And when glucose appears again, as you have a large amount, still a large amount of X4, and X4 is important for the first reaction to occur, because X4 is going to be the donor of the phosphate group to phosphorylate glucose into glucose 6-phosphate, then as you have a large amount of X4, the system will recover very rapidly. Okay, all the concentrations of the intermediates will recover in a small amount of time. While in the system without the regulatory interaction, what you see is that everyone goes down rapidly after glucose disappears, and when glucose appears again, they start to increase again, but very slowly, because you don't have X4, so this rate is going to be very slow. So all the recovery of the system is going to be too slow. So they argue that having this regulatory interaction is an advantage for this organism, especially if it lives in an environment where the glucose supply is not constant. So, for example, you could argue that this would not be an advantage for bacteria that only grows in the lab, because in the lab you are always giving the fresh media to the cell culture, but if wherever this bacteria lives in nature, it could be facing this problem of having some periods without nutrients available, and this interaction would allow the system to recover more quickly than the other. Okay. Any questions about this? So in this paper, they use two models, one that is slightly more complex and one that is more simple. And these make us think about how do we choose the level of complexity that we want to use in our modeling. So previously, I said that mathematical modeling was a must to study complex systems with a lot of components and interactions. That's true. But mathematical models can be also insightful for some models that are not that they are not complex in terms of number of components and interactions, but they have some patterns of interactions, some regulatory interactions that are not so easy to predict their behavior without having the computational simulation at hand to explore that behavior. So sometimes we have to make a decision if we want to approach a biological system, making a more simplified version of the biological system that we know is not going to be accurate. It's not going to make very precise or accurate predictions, but the qualitative behavior of the system is going to be similar to the real system. And being a model with less parameters, less equations, we are going to be able to do much more simulations and explore the parameter space more deeply, and that will increase our power to analyze the behavior of the system. But if you want to have a model that makes accurate predictions, so that is quantitatively accurate, for example, if you want to use that model to engineer, for example, a biotechnological application, then we want an accurate model. And for that, then we have to work with more complex models, with more parameters. And the thing is that the most difficult part there is going to be find the right set of parameters. Yeah, with normally those more smaller models, or sometimes people call it toy models, are very useful to characterize design principles. That is trying to understand why the system is designed in a particular way. And further in this course, we will examine several examples of using these toy models to explore design principles of biological systems. The predictions only agree qualitatively with the biological systems, but normally those toy models are similar to a lot of different biological systems in different species or in different cell types. And so you study that only toy model, but the results are applicable to a high number of instances of that biological system. While if you are working with a complex system, you are making accurate qualitative predictions, but those predictions are just for that particular system, because you have to have very exact parameters to be able to make those quantitative accurate predictions. This is a citation that is commonly referred when talking about modeling, is that we should keep in mind that all the mathematical models that we are going to use are always a simplification of the real system. So ultimately they are wrong. And that is a problem, for example, when you study a biological system with theoretical models. At some point you should interact with people doing the experiments with that system, because you need data to fit the parameters, or you want to test some new predictions that you made with the model. And usually when you interact with the people in the lab that works with the system, and when they look at your rules, at your equations, and they try to understand it, normally what they find very easily is flaws in the model. So normally, even if we are working with a very complex mathematical model, there will always be flaws. There will always be details of the real system that we are not including in the model. So there are always simplifications or assumptions that we do when we write the systems of differential equations to describe the system. And normally that's a problem, because the experimentalists get very suspicious of the results of the simulations, because they know that there are components of those systems that are not included in the mathematical description of the system. So the models are always wrong, but we want them to be sufficiently correct. So not absolutely correct, but sufficiently correct. And actually, when they are not correct, so when they fail their predictions, is when they are more informative, because they tell us that there is something that we have to change in the model. It's either the parameters or the components or the reactions that are included in the model. We have to change it to get a better model that makes better predictions. And we normally build a model to answer or to test a particular set of hypotheses. So even when studying the same system, the model has to be useful. So we can have different models for the same system, because we are answering different questions. Okay, some questions about this? No? No doubts about it? So for some of the students, they already listened to these presentations in another course. Sorry?
I said I'm fine, thank you.
Okay, thank you. Nice. Now, how do we build those mathematical expressions, so those differential equations. How do we translate those conceptual models to a set of equations? So the first thing that we have to do when we look at the conceptual module and try to write a system of differential equations to describe it is, from all the components that are in the conceptual model, one has to decide which one of them are going to be the dependent variables. And for sure, whenever you are trying to, as I said, you build the model to test, even that in virtually or in silico, but you want to test some hypotheses about the model, the system that you want to study. And whenever you have a hypotheses, you have also independent variables and dependent variables. So normally an hypothesis states that if I change something, a variable, I have an effect on other variable. So these outcome variables, the ones that are in the cause-effect relationship, the ones that suffer the effect, are going for sure to be dependent variables. Okay, so we want that in our model, the variables that we, in our hypotheses, are also dependent variables, should be dependent variables of our model. And then, of course, we want to make some in silico experiments where we change some, we make some perturbation to the system and see if the effect on the dependent variable is happening or not. So we want to have some independent variables that we can manipulate, that we can change each value, and that affect, in the, according to the model, have a chance of affecting the dependent variables. So at least you have to have some independent variables that you would like to manipulate, and some dependent variables that you are interested in studying their behavior. But in the connecting those independent variables and the dependent variables, probably you have a larger number of components that interact between them by regulatory interactions or by chemical reactions. So you need to choose which components are going to be either independent variables or dependent variables. And eventually there are some components in some conceptual models that you are simply going to ignore. Okay? You can ignore components either because in the time scale that you want to make your in silico experiments, you can assume that those values are constant, or for example, as that case of the ATP molecule is a component that participates in such a large number of processes that you are not going to include all those processes in the model. So you are not going to consider that variable. It's a constant. It can even disappear because its value is going to be hidden in the values of some parameters of the model. And of course, sometimes there are parts of the models that you are simply not interested in studying the behavior of those parts of the model. And if you can ignore the interactions of those parts of the models with the components that you are interested in, so looking again at the network of interactions, usually using the knowledge that we have been talking previously about the networks, if you identify, you have the conceptual model with all the components and interactions. You identify where are the interesting outcome variables, where are your interesting variables that you are going to perturb in your experiments, and then you try to identify the subgraph of that conceptual model that includes those interesting variables that you want to include in your model, and all the other stuff that is important to connect them. And if you find a highly connected subgraph that has a small number of connections to the outside, that is a nice way to define the frontier of your model. Whatever is outside, so you have very few connections to the other components, and you can isolate that component and work on that module and work with that model in a more dynamical, precise way. Okay. Sometimes those independent variables are just environment and environment variables, like the example I was talking about light intensity for the metabolism of plants. It could be temperature or other environmental variables that are important to define the rates or the processes that are working, that are happening in the system, even if they are not that interesting for your hypothesis. But if they are important to define the behavior of the system, you have to include them. And if they are they are important to define the environment within where your system is included. Then after if identifying which of the components, so in those conceptual models, normally you have the identity of the molecules or the proteins or genes that are involved in your model. Those are the components, and then normally you have arrows connecting them. And those arrows can represent different things. So you must be careful about interpreting what the arrow in the conceptual model means. Some of those arrows are mass fluxes, so they correspond to chemical reactions or transport of a component between different compartments of the model, so chemical or transport reactions. So each mass flux, each arrow that corresponds to a mass flux, it's going to have a rate expression. So you have to have a way to compute how quickly, so how many times per how many times per unit of time that process happens. So you have to identify all the mass fluxes, and all the mass fluxes will have a rate associated with them. It is important that those mass fluxes you identify where that arrow comes from and where does it go to. So it starts in one or more components and it ends in one or more components of the system, and that is important because that rate expression is going to appear in the differential equations of those components. It's going to appear with a negative sign in the differential equations of the components that are consumed by that reaction, and it's going to appear with a plus sign in the differential equations of the components that are formed by that reaction. Some chemical equations have more than one reagent and may have more than one product. Let me check if I can write here. Sorry. So sometimes you have a simple reaction. So you have one molecule that gives origin to another molecule. But it can be that you have A and B combined together to give origin. Oh, this should not be. A and C, for example, this is C, combined together to give origin to B. Okay, so and it is also possible that you have, for example, another product. So the arrows of mass fluxes could have multiple starting points and multiple endpoints. And sometimes you have to be careful because sometimes this is not well represented in the conceptual model. Another thing that is not well represented in conceptual models, sometimes that you have stoichiometric coefficients. So you need two molecules of A and one of C to give origin to a molecule of B and D. And knowing this is important because that is going to affect both the rate constant or the rate equation and how the rate expression is going to be introduced in the differential equation, because if two molecules of A give origin to D and B, then in the differential equation of A, and this, imagine that this is the V1 process that has a rate expression. In the differential equation of A, I'm going to have somewhere in the differential equation minus 2 times V1. Okay. And for example, while in the differential equation of B, I will have plus V1. Okay, so the stoichiometric coefficient is important to define the actual rate expression. We are going to see that later, and to define the coefficient of that rate expression within each differential equation for your system. Okay. And of course, you have to, sometimes these arrows are not so well organized because, for example, this same situation could be represented by, for example, C giving origin to D and C giving origin to B, while A gives origin to D, like this. So in this case, we have 4 arrows, but eventually they are representing just one chemical process. So you have to know if those 4 arrows are different independent chemical processes, different transformations, or they are part of the same chemical process, because each chemical process or transformation process has one rate expression and not multiple rate expressions. Okay. So you have to be careful about understanding what is the meaning of each arrow. So each arrow of mass flux will have a rate equation, and the stoichiometric coefficients may be even, so you have to be careful about that. The reactant stoichiometric coefficients will affect the expression for the rate value, and all the coefficients must be accounted when writing the differential equations. Also, sometimes in the conceptual model, the arrow is only represented in one direction, but the chemical process may be reversible. So in that case, sorry, in that case, you can have, so if you have a single arrow connecting A and B, you can decide that this is just a process, and if this is reversible, the expression of V1 will have 2 parts. One part that is the rate in the forward direction minus a rate in this is the forward direction and this is the reverse direction. Okay, so the rate expression will actually be a difference between 2 individual rate expressions, one in the forward direction and then in the reverse. But also you can do this in a different way. That is, you have A and B, and you have one arrow one way, one arrow the other way, and this would be V1, and this would be V2, for example. And then you have this would be V1, and this would be V2. So you can make a choice of how do you want to represent, just have one rate expression with the 2 fluxes, or 2 independent fluxes to compute the rate in both directions. Sometimes this may be helpful because you may already have, for example, a biochemical kinetics expression that includes in the same expression the both rates. That happens with some types of Michaelis-Menten equations for reversible reactions. So in that situation, it may be helpful to represent the 2 fluxes just in one equation, in one expression. Okay. That's what I said. But there are also in your conceptual models, usually there are other arrows that are regulatory interactions, and we can call those regulatory interactions, like instead of being a mass flux, is an information flux. And the thing is, those arrows are different because normally a mass flux, I need to write again. So if you have a mass flux, you have an arrow connecting A is transformed into B. So this is a mass flux. But if you have a regulatory flux, it's like C inhibits this process. So the regulatory interactions, if they are well represented, they connect one component to a mass flux arrow. Okay, so they do not end in another component. That means that this C is going to be important to the rate expression of this process. Okay, so this is a regulatory component, a regulatory interaction, while this is a mass interaction. It is helpful if, though, in the conceptual model, in that diagram, for example, the color or the type of lines are different between mass and the regulatory interactions. And there is also another problem, that is sometimes you have this arrow, and what people meant when they draw this conceptual diagram is that A activates B. How does this? How do we translate this into the more precise diagram that allows us to build the system of differential equations? So if A activates B, what can be happening is that you have B that is synthesized from some reactant, and B is degraded, and then you have A, and A activates the synthesis of B. Okay, sometimes this representation means this in actual terms. So you have to be careful to know if the arrows that you are looking at in your conceptual diagram are flux, mass flux arrows, or are regulatory interactions.
Could I ask a question? Because this regulatory interaction, is it something that just influences the reaction and doesn't participate or something in the reaction?
Yes, yes. So normally the regulatory the regulatory interactions mean that for example, this C is not going to, the concentration or the activity of C is not going to be affected by either this process or the fact that it regulates C. Of course, you can always model things in a more detailed way. For example, one way to represent these would be that you can have the conversion of A into B, but imagine that this is a slow process. But alternatively, you can have C that binds to A and gets a complex CA, and then CA gives origin to B and C again. So it should go like that. And this process now is a very rapid one. Okay, so you can model these in a more mechanistically molecular mechanism detailed way, or you can model these in a more conceptual or more simplified way.
Okay.
The regulatory interaction, sometimes it could mean that actually the regulator interacts physically with the reactant of the regulated process. but then it is regenerated. So in the total, after one cycle of these events, the activity of C is not changed.
Okay, makes sense.
Okay.
By the way, there's a question in the chat.
Oh, so
No, it just came, so.
Okay. Okay, the question is, yeah, I guess the question is, of course, these models can be applied at different hierarchical levels. I'm not sure if this is related to the last explanation about you can model these in different ways, but of course you can make these models to represent a lot of different processes. And as we said in the first lecture, all systems can be decomposed into subsystems, and all systems can be part of larger systems. And
Yes,
what
we have a question on BATPAP. I don't know if you see it.
but is that I'm
vou responder. Obrigada.
I was trying to answer it, right?
Sim, sim.
Okay. So the idea is that, of course, you can use these processes. For example, you can represent. Sorry. How do you can represent the synthesis of a protein just by a single step process, or you can represent it as a sequence of a long sequence of processes that correspond to the addition of each amino acid by the ribosome. So you can model these at very different levels of organization. But I'm not sure if I'm answering the question made by Luisa.
Yeah. Okay.
Yeah. Okay. So let me just take this out so that we can move on. If I close this now, I can. Yeah, I can. Okay. So you have to be careful about what the arrows mean, and if they are regulatory or mass fluxes, and if you have even information. Okay. That was what I was explaining. So some conceptual models have regulatory arrows between components, and that means that regulation is influencing either the synthesis or the degradation of the end component of the arrow. And normally when this happens is because we are looking at conceptual schemes of transcription regulatory networks or signaling pathways, and not of, for example, metabolic pathways. Another example that maybe I can, sorry. Yeah. Another example of those even fluxes is a different way is that, for example, A regulates the conversion of protein B to an activated state or an active conformation. So B with an asterisk, and of course there is a process that converts back B asterisk to the inactive form. And so A is so sometimes when you have A activates B, for example, in a signaling pathway, that may mean that A phosphorylates B and then B becomes active. That could be that A is converting B into the phosphorylated form of B that is active. And there is normally an hidden process in this scheme, that is, there is a rate of dephosphorylation that converts back B, the active form of B, into the inactive form. So you have to be careful about what are those arrows representing. If it is a phosphorylation, you can translate it into a model like this. But if it is a regulation of transcription, it would mean the other example that was you have B with the synthesis rate, and then A activates the synthesis. So these are different ways of that this single interaction can be representing either a signaling interaction or a transcriptional regulatory interaction. Okay. The other thing that you must be aware when you are trying to build a new model is if your model is a single compartment model or if it involves multiple compartments. This is especially important when you are modeling processes that occur in eukaryotic cells because they are highly compartmentalized. So you have different organelles in the cell, and the same molecule can exist in different organelles, and it must be transported across these organelles. And so the same molecule, if it exists in multiple compartments, that would mean that you have different variables in the model. So protein A exists in the cytosol and in the nucleus, then you have to have in your model a differential equation for A cytosolic and a differential equation for A nuclear. Okay? And of course, you have processes that are the transport of A across the two compartments. And if the two compartments do not have the same volume, then you have to be careful in designing those equations. Why? Again, I'm going to draw. Yeah. So if you have a cell, for example, like this, and then you have a compartment that is like this, and you have here a nuclear, and you have here a cytosolic, okay? And you have a transport reaction in one direction and in the reverse direction. So this would be the V1 and this would be V2. So to set up the model correctly, and usually the units of the model are in concentration units. So that means that you are modeling the variable A cytosolic is going to give you the number of moles of A per milliliter, for example. So it's a concentration unit. If you are building your model where the unit of each variable is the number of molecules, then you have no problem with this. But if it is concentration, you may have a problem. If the two compartments, like in this drawing, the nucleus is much smaller than the whole cell. Okay? And that is a property that changes from cell to cell, for example. And in this case, you must be careful because you must define the rates V1 and V2 using as a reference the concentration in one of the compartments. For example, V1 should be normally it would be a rate constant times the concentration of A in the cytosol. Okay? But each time the process V1 occurs, there is one molecule of A that goes away from the cytosol and appears in the nucleus. Okay, so it's the same molecule that goes out from one place and appears in the other, but the change in concentration in the cytosol, as it is much bigger, it's a smaller change in concentration than what you see in the nucleus, where that single molecule will affect in a smaller volume, will affect in a more intense way the concentration of a nuclear. Okay, so normally what we do is we have another constant. For example, you can see Kv, so it's constant related to the volume. That is the volume of the cytosol, for example, divided by the volume of the nucleus. Okay. And you define this rate expression using the concentrations of A cytosolic, that is the concentration in the cytosolic compartment. So where the change in concentration is smaller. So when you apply these into the differential equations of A nuclear, this will have a term that is plus V1. But the change in concentration that is described in this expression is the change in the cytosolic concentration. So in fact, in the nuclear concentration, this will have a much higher impact. So you have to multiply the V1 by the KV correcting factor. So because imagine if the cytosol is 10 times the size of the nucleus, then a change of 1 millimolar in the cytosolic concentration is going to give rise to a change in 10 millimolar in the nucleus. So you multiply it by this volume factor. While in the acytosolic differential equation, V1 appears without the correcting factor. Okay, so in which equation the correcting factor is applied depends on what are the concentration of the unit of concentration of the rate expression. Are they defined using as a reference the cytosolic compartment or the nuclear compartment? So you have to take care about that. There is also, I'm going to answer your question. Just let me say another thing. There is also another problem is that some reactions in cells take place in membranes. Okay, for example, there are some processes that occur only close to the membrane because the protein that does the process is a membrane protein. So here, this protein is converting some component into a different component. Okay, and this process occurs on a surface, not on a volume. And this is maybe also a problem when you model different cells that have a different relation between the area of their membranes and their volume. Okay, and this must also be corrected. And not all models take into account this. So there is a question.
Yeah, it's a technical question on biology itself. Does the nucleus have a lipid membrane as well to separate what is inside the cell from what is inside the nucleus, or does it work like by osmosis? Whatever is inside the cell is also inside the nucleus. How does it work?
So in eukaryotic cells, the nucleus is physically separated from the rest of the cell by a membrane, by a lipid bilayer membrane. So that is similar to the plasma membrane. So there is really a frontier. There are small molecules that are apolar or lipophilic that can cross those membranes. Those are either very small molecules or molecules that don't have polarity. So they interact very well with the lipids inside the membrane. But for most of the other molecules that are polar, for example, if you have alcohols or other molecules that have parts of the molecule that are slightly positive charge or negative charge, then they are difficultly, they cannot cross the membrane. They can only cross the membrane through channels or pores. So there are proteins within the membranes that act as facilitators of transports of stuff across compartments. They can be selective. They can select. I'm not. Is that the? Am I going in what?
You're going deep. You already answered my question. Thank you.
Okay. Okay. So yes. So normally these transport reactions are associated with the presence of proteins that specifically transport some molecules across, even proteins that migrate from proteins and nucleic acids that migrate from the nucleus to the cytosol, they need to go through larger protein machines that form pores in the nuclear membrane that allow the transport of larger macromolecules across those membranes.
You're talking about the pumps?
Actually, I was talking about nuclear pores that transport proteins across large proteins. You have pumps normally used, so some pumps are used to transport substance against their natural gradients of concentration.
Okay.
So, and then you use, normally cells have gradients of ion concentrations, so potassium and sodium normally have different concentrations within and outside of the cell or within compartments, and then you can use that gradient to force the transport of other substances that are moving against their gradient of concentration. But that is but to model that in a mathematical model, a transport that is, for example, carried on by a pump, it's just like simulating an enzyme catalyzed process. So it is using some kind of energy, for example, either from the gradient of the ion, or some transporters consume ATP to transport the molecules across the membranes, and you are going to model it just as a chemical reaction that transforms one component into other in different compartments. Okay, so be aware of if you are modeling transports across compartments, or if you are modeling processes that occur that are catalyzed in membranes, you may have to be careful about considering the relationship between membrane area and volume of the cell, or of the compartment, or the different volumes of the compartments that are being modeled. Okay. So once you have sorted out what are all the processes, if they are mass fluxes or regulatory fluxes, if they are transporting stuff across compartments or not, then you can enumerate all the processes, the mass fluxes that are happening in your system. For now, you can start by not worrying about what's the rate expression for each of that processes, but then you can right away write the differential equations, because each differential equation is the sum of all the mass fluxes that either produce or degrade the dependent variables. So if you have so this is a very simple process. If you have in your model, you have the component A that gives origin to some stuff through V1, is formed through V2, and for example, and is also transformed to other stuff by V3, and it's formed by V4. So A is a dependent variable, and these are all the mass fluxes that contribute to it. So the differential equation of A is going to be V2 plus V4 minus V1 minus V3. Okay, if some of these reactions, for example, if V2 produces, each time V2 happens, produces 2 molecules of A, this has to be multiplied by 2. Or the same thing, if V3 consumes more than 1 molecule of A, it has to be multiplied by the stoichiometric coefficient. But writing these equations is really simple, because you just make a list of all the processes of mass fluxes that affect that dependent variable, either producing or degrading, and you make this expression with sums and differences. You can be more organized, and in fact, you can say that every differential equation is a sum of all the fluxes with either a VI with multiplied by a stoichiometric coefficient of that process for that component, and with another product that is either a 1 or a -1, according if it is a flux that is producing A or degrading A. So you can write this in a general way. But you cannot forget the stoichiometric coefficients, and the sign is either plus or minus because it is either producing or degrading A. You do that for all the dependent equations, the dependent variables. Be sure that normally each process will appear at least in 2 differential equations, but it can appear in more if the reaction involves more than 2 molecules. It is important to have do not forget one rate value in one differential equation, because if you forget that, when you start modeling the system, there is going to be a variable that is either it's not going to be synthesized enough, or it's going to be degraded more than it should, and then you can get negative concentrations or concentrations that keep increasing until infinite, because you forgot to include one of the rate processes in your differential equation. So being sure that you include all the rate processes in all equations with the right stoichiometric coefficients ensures that you'll have a mass balance in your system. And that is important because otherwise you can have mass appearing in your system from nowhere or disappearing from your system to nowhere. Usually there are some fluxes, as biological systems normally are open systems. There are fluxes, there are mass fluxes that come from the outside of the system, and there are also mass fluxes that go to the outside of the system. So you have an arrow that comes from nothing and produces a component of the system, and some of your components have an arrow that degrades them out of the system, and you don't care what are they being converted to. And that allows you, normally that allows your system to achieve a steady state, because you have a flux of mass going in and you have a way to get mass out of the system, and your system will eventually achieve a dynamic equilibrium that is a steady state. Any question? So once you have the differential equation, then you need to fill in what are the values of all the rates. So all the V, V1, V2, V3 that you wrote in your equation, differential equations, what are the values for those rates? So the most common option is using mass action laws that come from chemical kinetics, although they are applied to very different systems. For example, now for sure you have heard about those models of the epidemics that are being used to predict the evolution of the COVID epidemics. They are based, the most classical ones, not all of them are based on differential equations, but the most classical ones are based on differential equations. And the conversion of individuals between the individuals in those models can be susceptible, infected, recovered, and you can have different compartments within each of those classes of individuals. And the conversions of one individual from a susceptible class to an infected class is modeled by a mass action law. For example, in those cases, let me write, in those epidemic models, what you have is, for example, you have an infected individual that randomly bumps into a susceptible individual, and what happens is like a reaction, chemical reaction, and then you get 2 infected individuals. because the one that was infected keeps infected, and the one that was susceptible is converted into an infected one. So the rate of this process is going to be a rate constant. If this has a V equals to a rate constant times the abundance of I times the abundance of individuals susceptible. So the rate mass action laws are always written like that. Each rate is the product of a constant, a rate constant, multiplied by the amount or concentration of the reactants of that process. So the ones that are in the origin of the arrow. And if you have more than one reactant, those reactants, there is a product of the abundance or concentration of those reactants. And if some of the reactants have participate more than one molecule of each in the reaction, each reactant is raised to a power that is the kinetic order, and this kinetic order is the stoichiometric coefficient with which that reactant participates in the reaction. So these mass action kinetic laws are accurate if you are working in a homogeneous environment. So your molecules are exist in an homogeneous space. There is no spatial structure. All the processes occur with the same probability in all the volumes of your space, or all the areas of your space. And it is valid for simple molecular reactions. If you have catalyzed reactions, eventually these mass action laws are not so accurate. But normally, biological systems are well represented or well approximated by dynamical systems using mass action laws. But still, there are some processes that mass action laws are not able to reproduce, and that's where enzyme kinetic rate expressions appear. Why? Because enzyme catalyzed processes that are very common in biological systems have a property that is they saturate. So if you have a process whose kinetics is described by a rate law of the kind of mass action. So if you have a rate that is influenced by A, if it is influenced by a mass action law, this is going to be a straight line, right? So the velocity of the process or the rate of the process will grow up to infinity. It will keep growing forever. But in a catalyzed process, that doesn't happen, because normally what happens is that if the amount of A is small, you have something like this. When you get to some point, the velocity will saturate, because there is no capacity for that process to happen at that increasing rate. But I said that most biological systems are well approximated by kinetic laws. That happens because most biological systems, when you go and make simulations about them, the rate of variation of A, for example, it's only. So most biological systems have some buffering or homeostatic properties. That means that they tend to converge to steady states, for example, or other kinds of stable states. And so the concentrations of some of those components do not vary a lot across time. So for example, if my A component, my A molecule, does not go below this level and does not go above this level, so all the variation in the rate of that process, depending on A, will always happen in this period. So this segment of this curve, that is an hyperbole, is well approximated by a linear segment. So that's why many times, although we know that some processes are catalyzed and they saturate, the rate saturates, within the range of concentrations of the reactants that happen in the system, it is not expectable to see the rate changing from this regime to this one here. So normally the rates behave within a limited range, so mass action law is able to approximate correctly or sufficiently accurately the rate of the process. But sometimes if you are in a situation where you think that due to the perturbations that you are going to apply to your system, eventually the concentration of the reactants is going to have a wider variation, then and that wider variation will span these regions where the rate increases very rapidly and the other regions where the rate will saturate, then it is advisable to use enzyme kinetic expression. So the most common one is going to be the Mqbs expression. This is the equation for an hyperbole that I draw here. So this hyperbole has this property that it will saturate for infinite concentrations of the substrate or the reactant, and there is going to be here it will saturate at a maximum value. In this case, that maximum value is given by this constant multiplied by the availability or the amount of the catalyzer, the enzyme. Sometimes it's represented by capital V, so it's the maximum velocity. And then there is a velocity here that is 1/2 of the maximum velocity that corresponds to a concentration of the reactant that is called the Mqbs constant, that is this K here. So this K is the concentration of A that gives a V that is half of the maximum velocity. So this is Km is and the V max are the main parameters of these enzyme kinetic expressions, but you have different variations of kinetic expressions. For example, if you are considering a reversible reaction, you have this more complex expression. If you have two substrates or two reactants, you have here the expression for a catalyzed reaction with two reactants, A and B, or you can have processes, enzyme catalyzed processes that are inhibited, and in this case, the inhibition is sorry, let me. The inhibition is carried away by this molecule M, and you have biochemical enzyme kinetics expressions for inhibitions. But most commonly, when you want to
Professor, you are muted.
Can you hear me now?
Yeah. Yes.
Yeah, they have that problem with the mouse that I cannot see yeah. Let me try to shut this down. Okay, but many times when you are trying to model processes that are regulated, you use what is called the Hill equations, that are the ones in below. I don't know if you are seeing my pointer, mouse pointer.
No.
No, because I also cannot see it. So the thing is that you can have Hill equations that are the ones in below, where where you have that, for example, in the right below, you have a Hill expression for an activation, where you have that m raised to v divided by km raised plus m raised to v. That is a activation Hill equation. And on the other side, on the left side, you have an inhibition Hill equation where you have that term km divided by km plus m raised to v. Those kinds of expressions give the shape of the rate in order to the modulator the shape of a sigmoid. Okay, let me try. I would like. Where is my mouse? Okay, are you listening to me?
Yes.
I would like to change the slide, but I'm not being able to change the slide. And it's 11 o'clock, so it's the end of the class. So probably we'll continue this next week. Any questions? No, so next week we'll end discussing these rate equations, particularly the Hill equations. So is there a question? The raised hand is the same as before, Luisa. Luisa is writing. Do you want to ask something?
I don't, thank you.
So the problem is I'm not being able to close the drawing tool, and therefore I'm not able to change the slide. But I'm happy to answer any question that you have while I'm trying to. No questions? Okay, so so next week we'll continue discussing these types of models and what you can do with these types of models.
Okay, thank you.
Thank you.
Thanks. See you next week.
Thank you.
See you all next week.
Thank you.
Thank you very much. See you next week.